Right. Assalamu alaikum wa rahmatullah wa barakatuh. Distinguished guests, esteemed colleagues, ladies and gentlemen, good afternoon and welcome to today's main event. I am Omar Kniou, Professor of Applied Mathematics and Computational Science, Chair of the 2018 Enrichment Program. It is my privilege to call on Dr. Yves Nanou, Vice President for Academic Affairs, to deliver a welcome address.
Thank you, Omar. Good afternoon. Salam alaikum. Good afternoon, Justin. So this year again, we have a great Fall Enrichment Program, and I would like, first of all, to thank Omar. Omar and Marie-Laure Boulot, you guys, you did a great job putting up a very attractive program and selecting the team that is related to marine biology, coastal biodiversity, and coral reefs, and so on, all topics investigated in this university, but also great concerns to laymen like me, but also address, in big media, right? So I think this is of interest to all of us, and this is we have 4 great speakers that have been invited, and I would like to take this opportunity to thank them. So today we have Luiz Rocha. So Luiz Rocha will be introduced by Omar later on, and he will have a keynote lecture on Twilight Zone. So I think that Omar will say more about Luiz Rocha. So tomorrow we will have Greg Skomal about sharks. I hate sharks. Now, each time I go to the beach, I just wonder whether they will have sharks and so on. We will learn about sharks and jaws and so on. And then Professor Catherine McFadden about biodiversity. And on Tuesday, late afternoon, we will have Dr. Gustave Paulet about how little we know about coastal biodiversity. This is also something that we study here. So without further ado, I would like to, besides our own students and faculty and leaders, besides them, I would like to extend a very warm welcome to the students and professors and colleagues from other universities. From King Abdulaziz University, we have invited them. They were kind enough to respond to our invitation. But they are also colleagues from other universities, from University of Jeddah, from Al-Qura University, Taif University. I don't know whether all of them are, but we extended this invitation. Hopefully the students, professors are here. We have 3 great days, 4 great speakers. Hopefully you will enjoy. Myself, I know little about this topic. I am a poor chemist, and hopefully the only thing I know about biodiversity is through CNN, through New York Times, so I think that I will learn a lot. So, Omar, the floor is yours to introduce our today's speakers. Thank you.
Thank you, Yves. It is my pleasure today to introduce today's speaker, Dr. Luiz Rocha, who is Curator of Ichthyology at the California Academy of Sciences in San Francisco. Luiz is a National Geographic explorer, marine biology professor, and has made numerous contributions, including more than 120 scientific articles and 1 book. Currently, Luiz's main area of work involves the exploration of the little-known mesoscopic 60 to 150-meter deep coral reefs throughout the tropics. Luiz's work has mainly focused on describing the uniqueness of the fauna from these depths and on advocating for both the inclusion of deep reefs into existing marine protected areas and the creation of conservation areas dedicated to those reefs. In addition to scientific contributions, his work has been featured in many popular media outlets, including the New York Times, Wired, National Geographic, Smithsonian Magazine, among others, and has supported conservation efforts across the globe. Luiz is certainly no stranger to the kingdom or to KAUST. He has led and participated in numerous expeditions around the world, including both ends of the Saudi Red Sea coastline. He has been a regular visitor to KAUST, helping in planning and designing biodiversity sampling in the Red Sea since 2012. Luiz, it's a pleasure to have you back at KAUST, and we are very much looking forward to your keynote.
Thank you. Thanks for having me. Pleasure to be here. Let's see if this goes back. Yes? Yeah, I mean, it's a pleasure to be here. I'm going to talk about, so historically, I haven't done much of this Twilight Zone mesophotic exploration much because I didn't have a lot of institutional support when I started my academic career. But now I'm finally in a place where everything aligned and I have the support I need to do this. Before I go into the actual talk, I wanted to thank my host, Tana Sinclair Taylor, Mike Barrowman for the invitation, Omar for the introduction. It's again great to be here, an honor to be presenting this at KAUST. This is something I really want to do here. I'm probably going to close the presentation with a little pitch to try to see if I can move this research line to the Red Sea. We've had some obstacles in trying to do that, but hopefully we'll overcome that over the next few months or years or so. So before we descend into the Twilight Zone, just to make sure that everyone is in the same page, that everyone knows what I'm talking about, I'll have to introduce you to a little bit of coral reef biology and how coral reefs function in general so that you can understand fully what I'll talk about when we get to the Twilight Zone. So when you think of coral reefs in general, that's what you think of. It's like super clear water, very shallow reef, very colorful, a lot of stony corals, high diversity, a lot of fish, a lot of invertebrates, and it's an ecosystem powered by the sun, more specifically by the relationship between zooxanthellae, between microscopic algae and coral, that allows the coral to grow and thrive. Now, if you've been paying attention to anything that's going on with coral reefs around the world, you know that coral reefs are in big trouble worldwide. And there's many reasons for that. So it seems like every week there's some sort of news article out there about coral reef decline. And you hear about this all the time. And there's many threats affecting coral reefs today, including overfishing, plastic pollution, just physical destruction of the reefs. When you go out on a boat and you throw an anchor, you break a lot of coral. Nutrient input, so when there's a lot of sewer going out from cities, for example, they cause algae to overgrow coral and outcompete the corals. That's very bad for the coral reefs too. But lately, the biggest problem by far has been this. It's climate-driven change that's causing coral reefs to bleach worldwide. So in the left here, you have a photo taken of a healthy reef in the Great Barrier Reef in December 2014. So what gives the color to the coral, so the reason why they look brown with the blue tips like that, is that relationship between the coral and the microscopic algae. There's a microscopic algae that lives within the coral, and they form what we call a mutualistic relationship. So it's a relationship when the two organisms benefit from that relationship. So the coral, which is an animal, feeds on the sugars that are produced by the algae, but the algae feeds on the waste from the coral. So both of them get something from the relationship. It's a mutualistic relationship, but it's a very strictly defined relationship that requires very specific conditions to occur. One of the conditions is a stable temperature where both organisms are used to. In some places, when the temperature rises above a certain level, the coral gets so stressed that they expel the algae. Now, when they expel the algae, that's what the center picture is showing there. The coral bleaches. So the coral in that center picture, it's still alive. It's white because all of the algae that was within its tissue was expelled. Now, if the temperature stays warm in that area for long enough, then the coral will eventually die, which is the last picture there. And it probably gets overgrown with algae. And this is very bad for the coral reef in general because it decreases diversity in the coral reef ecosystem as a whole. So there's a lot of different other organisms, from everything from invertebrates to fish to mammals, that depend on the basis of the relationship that's provided by the coral. The coral are the builders of the ecosystem, and everything else is kind of related to what the corals do. So if the corals are dying, everything else in the coral reef suffers. Now, that's been widespread. What's been in the news lately a lot is this, the Great Barrier Reef, which suffered tremendous bleaching over the past 2 or 3 years or so, repeatedly. In 2016, all of the red dots there that shows areas where more than 70% of the corals died along the Great Barrier Reef. And this is a 2,000-kilometer-long coral reef of Australia. In 2017, there was a mass bleaching event again, where most of the center of the Great Barrier Reef died. On the far right there, you notice that an area that escaped bleaching in both years was the far south of the Great Barrier Reef, so it's all green in both years. And that's because the further south you are in the Great Barrier Reef, the cooler the temperatures are. So the temperatures in the south didn't get warm enough to cause bleaching in either one of those years. But scientists are noticing that bleaching has got further and further south, not only in the Great Barrier Reef, but elsewhere. Now, this was what was in the news. If you Google coral reefs for the past 2 years, all about coral reef decline and coral reef bleaching and how climate change is affecting coral reefs. So that's the biggest global threat to coral reefs today, in addition to all of those other smaller threats that I talked about. Now, having said that, we can kind of move on to what's happening to the deeper reefs. So when I say Twilight Zone, or the technical term for its mesophotic coral ecosystem, if there's any oceanographer in the audience, when I say Twilight Zone, they're probably shivered a little bit, because there is an actual zone that the oceanographers define as the Twilight Zone that's much deeper than what I'll talk about. So the mesophotic coral ecosystem, which is nicknamed the Twilight Zone, is between, the official definition is between 30 meters and 150 meters depth. I think 30 meters is too shallow for it, so I personally define it between 60 meters and 150 meters. And that is an area of coral reef that there is still a lot of diversity. There's a lot of structure. The structure is mostly provided by corals that grew on that particular depth when the sea level was lower. So if you remember from your old oceanography and even Earth history classes, sea level goes up and down with glacial periods. So when there's a glaciation, the Earth as a whole cools off. The poles accumulate a lot of water from the ocean into the ice caps, and then the sea level drops. When the sea level drops, the coral grows with the sea level, where the sea level is. So if you imagine a sea level that's 100 meters lower than what it is today, the corals would be growing exactly at the depth where the Twilight Zone, where the mesophotic reefs are today. So when the sea level rises back to a level that it is today, where we are in a warm interglacial period, then all of that structure is left behind, and it provides homes for things, for organisms to live there, fish, invertebrates, and many diversity of organisms. So it's a coral reef-related fauna, but it's not necessarily the same fauna. Now, it has remained completely unexplored. I think it still is. I still consider it very much unexplored until today for a number of different reasons. It's very hard to get to. So if you think of scientists that are divers and go out sampling the coral reef, they use the normal scuba tank, just one scuba tank, one regulator, one BC, so the normal scuba gear that the same recreational divers use that doesn't require a lot of training. It's not very expensive, so it makes the shallow reef, that top portion there, the top 100 to 150 feet or 30 to 45 meters or so, it's very accessible to the normal scientist, the normal researcher that just go through a recreational scuba diving training. To get to those depths, to the Twilight Zone, you need technical scuba diving training, which is very hard to do. It's very expensive. The gear that you use to go there is much more expensive. Now, the alternative would be to study that with a submarine. But the submarine, then it's very, very expensive. To operate a submarine these days, it's on the tens of thousands of dollars a day to operate. So it's outside of everybody's budget. And if you do have that budget, when you have access to a submarine, you're not going to trash around between 60 and 150 meters. You go as deep as the submarine allows you to go, and you explore the other area, the deeper areas that are almost as unexplored as the Twilight Zone. Not to mention that my specialty happens to be fish. And sampling fish in the coral reef or studying fish in the coral reef with a submarine, I think it's equivalent to try to study birds in the rainforest with a helicopter. It's not the right tool for the job. The submarine is too noisy, it's too bright. When it gets close to the reef, you see every fish hiding. So why don't scientists just do the technical diving and use technical gear and do it? There's a few reasons. First one is this. We have to breathe. It's so deep that we have to breathe different gases, and we have to carry a lot of gas. The deeper you go, the more gas you need to fuel your lungs because of the physics of water. It compresses the gas, so you need a lot of gas. You need the multiple tanks. You need different mixes of gas. Both oxygen and nitrogen, which are the gases we breathe in the atmosphere, they become toxic at depth. So we have to replace that with helium. We mostly breathe helium when we go really deep. And helium is a very expensive gas. So much so that for a dive like that, this guy did on the left here, he spent maybe $2,000 on helium alone. So we're at 150 meters depth, breathing from one of those tanks, it could be as much as $10 a breath, just from helium. So people stayed out of this area because of this. Now we have a more relatively recent technological advancement that allows us to go to those depths more efficiently, which is the rebreather. So that's what this diver is using here. It's a much more compact system, and it allows you, so that rebreather with just those two little tanks on the back there, allows you to stay up to 9 hours underwater. So with that, we can actually go there and save a lot of money on helium and do this in a much more effective way. So how does that rebreather work? Why does it save us so much money? In essence, it allows us to breathe the same helium. So that helium that costs us $10 a breath, it allows us to breathe the same helium over and over again. So because it's a rebreather, when you exhale, it's not like a normal tank. When you exhale with a normal scuba diving gear, the bubbles come out. When you exhale with a rebreather, your exhale comes back into the system. Then there's a filter in the back that removes the CO2, so the byproduct of respiration that is toxic, CO2, carbon dioxide. The filter in the back removes it. So let me go back really quickly to the other slide here. That's the filter in the back is behind that black box there, but you can kind of see it. It's the white material inside the clear, transparent bucket. That's the filter that removes the CO2 from the mix. And then there's 3 electronic oxygen sensors that add oxygen only as you need it. So the oxygen, when we breathe in and out, depending on how much exercise we're doing, we never actually use all of the oxygen that we breathe. We always exhale oxygen. So we just need a tiny bit of oxygen added every few breaths to keep the oxygen at a pressure that maintains us alive. So this electronic system adds oxygen only as we need it, and then the same helium comes back without CO2 and with the oxygen you need to breathe. So because it's such an efficient piece of gear, it allows us to stay 7, 8, 9 hours in the water. The limitation there is the filter, not the gas itself. And because we're breathing the same helium, it's quite cheap to operate even in long field trips. So now that we have access to all of this, and we went through all of the training, we are going through locations around the world, trying to look at what's happening in those deeper mesophotic reefs. And one of the first things we wanted to test as part of my research program at the Academy was to try to figure out if those deeper reefs are a refuge or not. So there's 2 big assumptions that are pervasive in the literature about mesophotic coral reefs. So every paper you look at about mesophotic, deeper Twilight Zone coral reefs, they start with a sentence saying that those reefs are important because they might be a refuge to the shallow coral reef fauna. So if you go back to the start of the talk, coral reefs are declining everywhere. These deeper reefs are an extension of the coral reefs. They're inaccessible or very little accessible to scientists. So everybody assumed that the same fauna that was in the shallow reefs extended to those deep reefs, and because they are inaccessible, they might be a refuge or a safe haven for the shallow coral reef fauna. Now, that assumption that they're a refuge reigns in those two statements there. The first one is that there should be a lot of overlap between species in the shallow and the deep reef. And the second one is that those deep reefs are outside of impact, other impacts, including everything growing from climate change to overfishing to plastic pollution to everything, should be less impactful in those deeper reefs. So two things. One, they have to be overlapping. Two, they have to be kind of sheltered from those impacts. So the first thing we did when we had access to the technology was try to test those two assumptions to see if those two assumptions were actually right. Now, what are those assumptions based on to begin with? The overlap is based on depth ranges. So if you go to a fish database, and I'm going to give you fish examples here because I'm a fish person, but if you go to any animal database out there, a crustacean database, you search the literature and you find a thing that we call the depth range. So it's the depths that all of those species were collected. So in this case here, this is the orange line triggerfish, Ballistopus undulatus. And there's this database online called FishBase. And if you go to the FishBase database, right in the bottom here, it says the depth range of this species is between 2 and 50 meters. So the way that people traditionally calculate overlap between the fauna of shallow and deeper reefs is by taking this number, putting it in a spreadsheet, and then because it goes through that first boundary, so the 0 to 30 meters is what's considered shallow reef, and 30 to 150 meters is deep Twilight Zone reef, that species would go in the database as a shallow water species that uses the mesophotic reef as a refuge. But if we look at record by record, what that actually means, we can see that 99.3% of all of the records for that species are actually between 5 and 20 meters. So the vast majority of the population of that species lives shallow reefs. This is an even more interesting case. This is another species, Studentis flavaguttatus, and the official depth range is 18 to 63 meters. So again, it goes in the database as a species that is a shallow reef species that uses the mesophotic reef as a refuge, just because the depth range spreads throughout that range. But when you look at where that species was actually collected, 99.8% of it is only mesophotic. So in reality, that species is a mesophotic species that once or twice was found in a shallow reef. And because it was found in a shallow reef, the depth range is, the official depth range is that, but that's because it's a collection of all of its depth ranges. But the place where you actually find it in the wild, it's not that. So it's definitely not a shallow species that uses the mesophotic reef as a refuge. Now, it's the same for every other organism out there. The same for corals. These are I think these are the two most endangered species of coral in the world. They're the two Caribbean acroporas, Acropora palmata and Acropora cervicornis. They declined by more than 99% in some locations across the Caribbean, not even because of global warming. They declined much earlier than that because of a different set of reasons. But 99.5% of their population is found between 0 and 10 meters, but their official depth range is 1 to 60 meters. So in one museum somewhere, one researcher once collected an Acropora palmata at 60 meters depth. It might have been a branch that broke during a hurricane and then kind of tumbled down the slope. The official depth range is that, but if you go into the field and you actually look at it, the actual distribution is not that. So there's all of those cases where they go into the database as being shallow species that use the deep reef, but that's not really what we see. Now, the other assumption that they are out of reach is simply based on this. It's based on the fact that we don't know what's there. So if we don't know what's there, we assume that there's no impact. So when we went out, actually in the field, using all of our complex rebreather gear, all of the training we had, we actually did counts. So instead of using the databases out there with the depth ranges, we actually did transects. We counted what was there. So we counted more than 50,000 fishes in 4 different locations, both in the Pacific and the Atlantic. We did photo credits in the Atlantic. Quite a few people are relatively comfortable with the Atlantic coral taxonomy, but not so much with Indo-Pacific. So we didn't use photos to look at Indo-Pacific coral distributions, but we did for the Atlantic. And we used what we were seeing in the field, and we kind of compared it with what we saw in what we obtained from those depth range databases. And we found a very different result. Now, the first thing we found was that deeper fauna is very different. So different that there's new species everywhere. And I'm talking about fish here. I mean, the people that work with invertebrates here know that fish are the best known, in terms of taxonomy, the best known group in a coral reef in general, maybe not even second to corals, because corals are a complete mess. But fishes are very well known in terms of taxonomy. People know them very well. People know what species they are because they're highly visible, they're easy to collect, and they're very different between each other. And every time we go deep, we find new species of fish, new species after new species after new species. This one is one we found in the Philippines. This is the deepest species of fish that was ever described based on a specimen that was collected by diving. We collected this at 153 meters depth in the Philippines. Anything collected deeper than that was collected by a submarine or an ROV or dredging or hook and line. So this is the deepest fish that was ever described based on a fish that was collected by a diver. And we find them everywhere. This is another new species of chromis. We have a manuscript in preparation right now, submitting the new species description. We have about 30 new species in our desks. This one is from Easter Island, a place that we didn't expect to find any new species because it's a very well-known fauna and it's very isolated. It's very impoverished compared to other locations in the Pacific. So we compare the species diversity in Easter Island to that in the Philippines, it's staggeringly different. There's only about 100 species of reef fish in Easter Island. They've been studied for 40 years or so. In the Philippines, there's more than 2,000 species of reef fish. So you expect to find new species in the Philippines. You don't expect to find many in Easter Island. We found not one, not two, but we find 4 new species of fish in Easter Island, including this one that we think is a new genus. So even goes beyond small, trivial differences into very large differences that make us classify them in different genera. Now, we generated this little table here just to show what we saw in terms of novelty. The expeditions in yellow there are the expeditions led by Rich Pyle, which is a good collaborator of mine and the pioneer in this kind of study of deep mesophotic ecosystem studies. And in white there are the expeditions led by the Academy to the Philippines, Vanuatu, Ponape, and Easter Island. So those are the new species we found. In Fiji, there's the 2 asterisks there because that particular expedition, they managed to use rotenone, a little chemical that you can kind of, some places you're allowed to use it to collect more fish than usual. Let's put it that way. And so they collected a lot more fish there because of the chemical help they had. We didn't have that chemical help in the other location, so it was lower. But still, this is the time of exploration. So this is the time we spent actually exploring those depths. And this is the little number we like to come up with. This is the number of new species we collected per hour of diving in each one of those locations. So even at Easter Island, it's 4 new species per hour of exploration. So the more we explore those depths, the more new species we find. And it's not just new species, it's also new records. So these are all new records from the Philippines. So these fish had never been recorded in the Philippines before. We did 2 or 3 expeditions to the Philippines. We recorded all of these species. Some of them were only recorded from Australia, some of them only from New Caledonia, some of them only from Japan. And we also found them that they have this really wide distribution. And the reason why they're not recorded in other places is because nobody went there and dove deep to see what was actually there. So it's a mix between new species and new records. And that generated this. So the numbers there, they show the dissimilarity between shallow and upper mesophotic. So that top 30 to 60 meters, the middle mesophotic 60 to 90 meters, and 90 to 150 meters we call the lower mesophotic. So the difference between those areas varies between 87.6 to 99.7%. It's very, very different. If we convert that into overlap, which is the first measure I showed, the overlap based on depth ranges is between 66 to 75%. The overlap based on transects, so based on what we actually recorded by looking at those depths, is between 5 and 15%. So it's a lot lower overlap. They're not the same community. Now, that kind of refuted that first hypothesis. We published that recently. If you're interested, I can send you the paper. And then we went to Guam. And in Guam was the place that I saw this more vividly, that those deeper reefs are not really a refuge. So these 4 photos here, they kind of give you a sense of what the mesophotic reefs of Guam look like. It's a gorgonian-dominated area. There's sponges, there's other encrusting organisms, there's a few corals, there's a lot of different fish. But by and large, the fish community and the invertebrate community looks nothing like the shallow community. Not only that, but I didn't see any shallow community whatsoever in Guam. All of the reefs were as destroyed as I've ever seen. Every time I go into one of these expeditions, that by the end of it, I'm super excited about all the photos I took, and I'm always hopeful for the future of coral reefs because we see pockets of places where corals are surviving. Guam was the first place I went to where I did not see that. All of the shallow coral reefs we went to 7 or 8 locations. They were completely, like, it almost looked like they were bombed. The interesting thing about those is that we did not see any of the shallow species in the deeper reefs. So we saw a relatively healthy, deeper mesophotic community that was somewhat sheltered from the impacts of the shallow reef, but we didn't see any of the shallow species surviving on those deep reefs. So in this particular case for Guam, even though it's slightly sheltered from the shallow reefs, or from the shallow impacts, there's no overlap, so the communities are really different. Is someone timing me somewhere? Can someone kind of wave if I'm running over time or something? Okay, because I can't see the time here, so I have no idea what's going on. All right, so, and sometimes I tend to talk too much. I get excited with this stuff. So, what about the impacts? In Guam, they were kind of relatively sheltered, but what about other locations? So we were very lucky in some ways. It was luck for us, but not lucky for the Bahamas. But we had an expedition scheduled for the Bahamas for months. We went there in October 2016, and we almost had to cancel the trip because there was a hurricane barreling down and went right through the Bahamas, exactly through the route of the cruise we were going to take. We were on board a liveaboard ship, and we went down the Bahamas chain to study. At the time, we wanted to study the impacts of an invasive lionfish in the Caribbean. Now, because of the hurricane, this Hurricane Matthew passed through the Bahamas just 2 days before we went into this expedition. We kind of switched gears, and we shifted from a lionfish expedition to looking at impacts of hurricanes into those deeper reefs. So a lot of people know, everybody knows that when there's a hurricane that passes, it causes waves, it steers up the bottom, and it really impacts the shallow reefs. And that doesn't happen here in the Red Sea. It's a very benign region, but it happens a lot in the Caribbean. It happens in the Great Barrier Reef with cyclones. It happens in the North Pacific with typhoons. It's a common occurrence, and the shallow reefs, they're kind of adapted to it because it's a local impact. It almost causes reefs to become a little more diverse. But everybody, because the deeper reefs are deeper, everybody assumed that the waves generated by the hurricanes didn't have much effect into those deeper reefs. So this was one of the first expeditions where our team had access to deeper reefs just a few days after the hurricane had passed, and we took the opportunity to look at impacts of hurricanes on those reefs. And this is what we saw. This picture was taken at 145 meters depth, and the reef is completely covered with sand. It's not supposed to be white like that. It's supposed to be colorful like the ones from Guam that I just showed you before. So it's just smothered with sand. The sand that was steered up by the hurricanes in the shallow simply went down through the slope of the reef like a cascade and then buried all the corals on its way down. It wasn't only that. We saw, not only in the Bahamas, but a lot of other places, we saw bleaching. So all of those pictures were taken deeper than 80 meters or so even in the middle to lower mesophotic, and we saw bleaching, we saw coral disease, we saw a lot of the impacts that are affecting shallow reefs. We also saw and documented those in the deeper reefs in the Bahamas and elsewhere in the Caribbean and in the Indo-Pacific. And we see these in a lot of different places. So this is my deepest dive ever. I was at about 153 meters depth in the Philippines, and I saw this big blue blob in the distance. I thought it was a sponge. So I decided to swim to it to take a picture. When I got close, it was this. This is a piece of tarp or plastic bag or something that is attached to a fishing line. So that's a fishing line there in the bottom. And this is the picture that we saw in the Philippines over and over again. It's the same amount of trash that we saw in the shallows, we saw in the deeper reefs. So trash is affecting the slope regardless of depth. This is another representative that was taken that same trip. So this is a type of sea urchin that camouflages itself with its surroundings. So if it's in a kind of seagrass area, it grabs a little piece of seagrass and attaches to it to look like seagrass. So they grab whatever is around them to camouflage themselves. And this one is doing it very well because there was a lot of plastic there, and this blue thing is a piece of plastic attached to it. And then it's hanging again on a fishing line. And this is 125 meters depth, also in the Philippines. Even in places that are relatively well protected. This was taken in Palau, in the middle of a marine reserve, and this is an anchor line from a boat that on its way. Now it's almost serving as habitat. There's a lot of coral growing in it, but on top of it, we see the destruction it caused to all of the gorgonians that it knocked over when it was actively moving around with it attached to the boat. And yeah, we see impacts like that everywhere. So now I'll tell you a little bit about one of the latest expeditions we did. This one was to St. Paul's Rocks off of Brazil. This is a part of the Brazilian territory, but it's very far from the Brazilian coast. It's about 1,000 kilometers offshore, so it's very far from shore. And this is maybe about 75% of the island. So if I were to turn around and take a picture pointing the other way, it would be all blue. It's in the middle of the ocean. That's smaller than maybe the size of this building. It's the whole island. Tiny, tiny little island in the middle of the Atlantic. So there's no support for diving there. The only reason why we were able to do it was because they used this ship, the Aleutian, and it had all of the support we needed for our deep diving. They could carry our helium. There was a decompression chamber on board, so we had a lot of support. We could do all of our deep diving there. We did our usual sampling. We went down with our rebreathers. It was the sharpest thermocline I've ever felt in my life. So usually when we're going down to those depths, we cross what we call thermoclines. They're sharp differences in temperature. Different water masses have different temperatures. So in the shallow waters, in the tropics, I'm a wimp. I don't like cold water. So I never travel to places where the surface water is less than 28 degrees Celsius. That's my limit. That's it. Yeah, I'm a wimp, I told you. So the surface water in St. Paul's Rocks was 28 degrees Celsius. We had these little 3-millimeter wetsuits. And then when we got to 60 meters there, the water drops to 12 degrees Celsius. It was freezing. The time to go up was when our hands went numb. So the hands go numb, let's go up, let's go up. But it was like a mix of crazy water masses. And then below this thermocline, the fauna was completely different. Everything in the bottom looked different, from the sponges to the fish to everything. And I have a little video here that kind of summarizes what we saw there. So this, I'm going to try to talk over it, but I'll let you guys listen to the audio in it too, because it's very funny. So one of the side effects, so helium allows us to breathe at depth, but one of the side effects of helium, if you've ever been to somewhere, a party or something where they breathe from helium from the balloons, you know how you speak, like with the helium voice. So that's what you're going to hear here. But that's, so I'm I'm the bottom diver there. Hudson is a postdoc at the academy. He's the top diver here. And Meridius, our dive safety officer, is the guy that is filming. The first thing he's going to show you is the depth. So let me let this go here, and I'll kind of try to pause it. So that's the depth there. That's 419 feet. That's about 118 meters depth. And we had both Hudson and I had just saw a most spectacular fish we've ever seen. We had never collected it before. We knew it was a new species immediately. So one of the things that it's really exciting about diving in those depths is like you feel like almost like an astronaut or a first person that goes in a location ever. Because when we talk about new species, a lot of the times people assume that we're talking about something that looks very similar to what we already know, and then we have to sequence the DNA to make sure it's new. For those guys, for the deep ones, they're so different that we know immediately, just looking at them, that they're different. And this was one of those cases. The fish was so spectacularly looking, so different that we knew immediately that was the case. So this is at the start of the dive, and we already have like 150 feet stop to do. That's 1 minute into the dive. So that's Meridius right there. He is filming. I'm collecting that crazy-looking fish. Hudson is collecting the fish. So I'm trying to get the fish out of the hole there. This little friend comes to say hi. Oh, the audio is not playing. Oh, no. Oh, if you were hearing the audio, Mauritius would be saying, look at the shark, with this helium voice. There's no way we can put the audio in there? It's not working? Okay, all right. So Mauritius is saying, look at the shark, and I'm saying, leave me alone, I'm collecting this fish. I don't want to look at the shark. And then the shark comes around. So I'm right below the shark at this point. The shark comes around and goes right on top of Hudson. So Hudson is there collecting the fish. Mauritius is screaming, look at the shark, look at the shark. Hudson looks at Mauritius, but not at the shark. He says, no, leave me alone. Don't leave me alone. I don't want to see the shark. I'm collecting the fish. So neither I nor Hudson saw the shark. Neither one of us saw the shark. And so this almost summarizes, it exemplifies everything I said so far. It's the uniqueness of the fauna represented by the new species and the impacts. So this is a 6-gill shark. It's something you would never see shallow in the tropics. They don't particularly like warm water. They're the opposite of me. They like the cold water. And that shark, you see that little line out of his mouth? It's a fishing line. So the shark that probably nobody ever saw before, well, definitely nobody ever seen this particular individual before, already has a fishing line attached to his mouth. And yeah, it's documented right there. So the novelty, the uniqueness, and the impact all together in the same video. Now, I'll let the video finish. Actually, I'm going to skip it to the next slide. It's a pity we couldn't hear the audio, but it was funny. But this is the fish we were collecting. That's the reason why we completely ignored that massive shark. We called it Sonorous Aphrodite in honor of the Greek goddess Aphrodite. And it got a lot of press. I mean, the press was fascinated by it. Everybody talked about it. About half of the articles about it are because of the name and the look of the fish, and then the other half is because we ignored the shark. So some of them show the video, others don't, but the whole press was completely stunned and captivated by it. I think the last time I looked at, there's 40 different articles about it throughout the world. There's a lot in Portuguese because we collected this in Brazil. We also talked about it to Brazilian reporters. So even something as basic as basic science-like taxonomy still gathers a lot of public interest. It is something that we can use to call attention to the existence of these habitats and to how important and how unique they are. This is the other side of the coin. So remember, I told you that island is the size of this building. The permanent presence there is 4 Brazilian researchers that the Navy rotates. Every month, they bring 4 scientists there, and then a month later, they come back and pick them up. And the reason why they do that and don't keep a military presence there is because it's a lot cheaper. Scientists are free. So I'll go there. They go there. Everybody goes there for free, and they stay there for a month. And then when you have a permanent presence in an island in the middle of nowhere like that, you get the rights for the exclusive economic zone around it. So that's the only reason why that research station is there. And it's manned, but it's manned by scientists because the Navy doesn't want to spend any money with military there. So they do the transporting, but they don't keep their crew there. But even though it's a tiny island, the size of this building, with 4 people living in it, I took all of these pictures there in the 2-week time we went there. I don't even know what that bottom right thing is there. It's a T-shirt. I don't know. I don't want to know. But there's a pipe there. There's fishing line and anchor line everywhere. This white pipe here is one of the pipes that they used to put fishing line to. So the whole fishing line probably went down the slope. Amazingly, you see clear impacts of human footprint in an island in the middle of nowhere with 4 people living in it. So it's just staggering. So what's the take-home message from all of this? Is the Twilight Zone or the mesophotic coral reefs a refuge? No, absolutely not. They're very different communities. There's very little overlap between them. 87.6 to 99.7% dissimilarity. And those mesophotic coral ecosystems, they are almost as impacted as the shallow reefs above them. So what are we doing about this? We're trying to raise awareness, and it's very hard. When you try to raise awareness, when you try to create a marine protected area in a coral reef, it's relatively easy because it's a very visually appealing, a very beautiful ecosystem. And you take anybody on a boat, you put a snorkel on them, you put them in a reef, they're going to be a coral reef conservationist like overnight. Because there's no way you show a coral reef to a person, they're not going to want to preserve it. But for the Twilight Zone, it's very different because that's what they look like to most people. It's out of reach, it's out of sight, nobody knows where they are, nobody knows what's there. And at the end of the day, people protect what they love, but they don't really even know that the Twilight Zone, the mesophotic reefs exist. So one of the things we're trying to do at the Academy, where I'm at, is we have this Hope for Reefs initiative that is a combination of research and exploration centered around those mesophotic reefs, but also looking at restoration of shallow reefs and also trying to advocate for expanding marine protected areas to include those deeper reefs. A lot of the times, the MPAs that are created in the Philippines and Indonesia and a lot of places that we go to, they're very small and they don't include the mesophotic reefs. So those are some of the places where we saw the most impact to the mesophotic reefs because the fishermen, they're kept out of the small protected areas, but they like to be close to the border. And in these cases, the border is the deeper reefs. So their lines are everywhere in the deeper reefs because they're forced to be just outside of the shallow reefs and those deeper reefs. Nobody knows they're there, the fishermen know they're there, but nobody else does, so they're not included on those deeper reefs. The other way we're trying to kind of keep track and maybe raise awareness that these Twilight Zone reefs exist is through an exhibit at the Academy. So this is probably a topic for a completely different talk, but this is an aquarium that we built at the Academy. It's one of 10 tanks. We have about 10, 12 tanks with just mesophotic ecosystem, Twilight Zone, fishes and other organisms, all of those corals, the gorgonians, the wire corals, everything you're seeing there is from the mesophotic, from Twilight Zone reefs in the Philippines, except for the tiny little pink fish there, but the big Neapolitan fish here is from the Twilight Zone in the Philippines. And we've even developed a decompression chamber. So if you ever went deep sea fishing, you know that when you bring a fish up from the deep, their swim bladder expands and they die on the way up most of the time because the swim bladder expands and then it pushes all of the organs out of the way and it kills the fish, basically. So we go diving the same way we collect the fish. We would be collecting them for science, but instead of putting them in a jar, we put them in a decompression chamber and we keep them alive and we bring them back to the building and we have this entire exhibit dedicated to this fauna to show people that they exist even and how marvelous, how beautiful, how different they are. In the same exhibit, we also talk a lot about how we access those depths and what we use to access those depths. So that's a fully dressed mannequin there with the actual gear that we use. That wetsuit is actually a used wetsuit. So when the exhibits department was making the exhibit, they were buying brand new gear, and I'm like, no, you're not going to put the brand new wetsuit in display. Take this one. So I gave my worst one that I had, and then I got the new one for me, and that's what they put in the in the exhibit there. So everything else is the same. There's a camera hanging. You can't really see it, but the camera is a housing that we flooded. We flood cameras constantly because their depth rating don't go as deep as we go. The scooter is the same thing, it flooded. So it's all a recycled, flooded or used gear that we kind of ended up putting there, and then we used all the new gear that they bought. So what about the Red Sea? What do we know about this? This picture I took here yesterday in a dive with Tane. We went out to, what is it, Shib Nazar? Shib Nazar. And that is like a tiny little taste of the Twilight Zone. I didn't have the proper equipment to do it. So this was taken at about 35, 38 meters depth. So it's that upper mesophotic zone under a ledge. So it looks different and dark like that because it was under a ledge and the sun was on the other side. So it looks a lot deeper than what it actually is. But the Red Sea in general is at the same time well-known and very poorly known when it comes to mesophotic. So this is a website called mesophotic.org, and it's run by Pim Bungarts, one of the researchers at the Academy. He works on corals. And he has this very complete database with every paper published about mesophotic ecosystems in the past 30 or 40 years or so, including historical things done with submarines. And the circles there, they represent the number of publications of mesophotic publications done in the Red Sea. This is not in the world context. We'll talk about that when we have the workshop the next few days or so. But this is just to show you what's known about the Red Sea, and you can clearly see that Israel is far way in ahead of everybody else. So almost everything we know, there's about 45 papers published about mesophotic coral reefs in the Red Sea. 42 of them are done in 14 kilometers stretch of Israel coastline. Everything else, so the other 3 papers, there's 2 or 3 papers done in Egypt and Sudan 30 or 40 years ago with a submarine, and there's 1 paper done here by Chris Foster's group and Marin and their colleagues. They looked at depth acclimatization, again, trying to test that idea that some species might use those deeper depths as a refuge. And yes, some of them might, but not at an entire ecosystem level. So even though there's little overlap, there's some overlap, and there is a potential for refuge, but not for enough to cause everybody to say that the only importance of those deeper reefs are because they're a refuge. So I think, I'm almost consider this kind of point here as a crossroads. I think KAUST could become the world leader in this type of research. So right now, the world leader in coral reef, mesophotic coral reef research is Hawaii. The second most publications are coming out of this Israeli lab. And the reason why I think the KAUST could be the world leader in it is this. This was the dive we did yesterday. And conditions, they do not get any better than this. It's impossible to get any better. We have that my 28 degree temperature is met above and beyond here. So we have nice temperature. We can spend 8-9 hours in the water. So when we go to something I didn't talk too much about is when we go to those depths, we accumulate so much gas. If you're a diver, you know that. But you accumulate so much gas going to 150 meters that you take a long time to decompress coming up. So we sample the fish for about 10 to 15 minutes, and if we spend 10 to 15 minutes at 150 meters depth, it's a 6-hour dive. So we spend about 6 hours to come up. That's why I like the warm water. I do not want to be 6 hours in a 25 degree, 24 degree water. I want to be toasty. And this is what happens here in Hawaii. The reason why they have the most number of publications there is because they have the funding, but they do not have these conditions. I've spent 3 years in Hawaii. Not one day was like this ever when I was there. So even though there's a lot of people working on mesophotic ecosystem there, they're limited by local conditions and by the fact also that the mesophotic reefs are kind of relatively far from where they need to be. And here we can get to a place like this in a 25-minute, half-an-hour boat ride. So, and the logistics are here. You have the boats, you have the marina, you have the dive thing. You have a chance to make this like the beacon of mesophotic coral reef in the world, if you want to, if you really want to. And with that, I want to say shukran, thank you for the attention, and I want to open up for questions. There's a lot of people that help me doing this. I'm not doing this alone by a long stretch of the imagination. Yeah, I'll be glad to answer questions if you have any. Questions? Yes, go ahead. Okay, so the question was why people don't use robotics or underwater cameras. They do, just not the fish people. Because they scare the fish away. So there's one method that people are using for fish that is working relatively well. It's called baited REFs. What does it stand for? Baited Remote Underwater Visual System. But it's a camera attached to a little square-shaped structure that makes the camera come down to the bottom and stay upright. And then they have a little bait in front of it that attracts the fish, and you can record and see what fish are there. Now, that's great if you just want to see what fish are there, but if you want to describe them, if you want to put names on them, you need the actual specimen. So I think that, to me, that's the most frustrating thing you can do. You just send a camera down and you see what's there, but you can't touch it, you can't go there, you can't see it with your own eyes. And then for the ROVs, they scare the fish, just like the submarine does. So you can do a lot of Coral work with the ROV. If it's a big ROV that has manipulators, you can even try to collect some corals. But those, they become very expensive too, and they require a much bigger platform than just the diving. So for fish, the best option is still diving. But for other things, there's a solution here and there, but not ideal. Diving is a lot better, a lot easier. And when I pick up things, so I don't know anything about nudibranchs, for example. I can't tell their families apart. But there's a specialist in nudibranchs at the academy, so every now and then when he's on an expedition with us, I just randomly pick nudibranchs. Half of what I collected in terms of nudibranchs are new species, and without knowing what they are. So there was another question on the back. Yeah.
Thanks for the talk. I think everyone's quite envious of you being able to go down there to those depths, or at least to places where no one's been before.
Right, right.
I was wondering about the effects of the hurricane that you saw in the Bahamas.
Mm-hmm.
Have you had the chance to go back or get at least photo footage of the area?
Not yet.
And the other thing, maybe, have you been to reefs in the Caribbean that have been impacted by hurricanes, but that were a longer time ago?
Right,
so
right.
What I'm wondering about is if that sand goes somewhere.
Right. I think it eventually gets flushed out, but not as quickly as in the shallow reef. So in the shallow reef, there's more energy, more current. It gets flushed out. Another impact that I did not talk about, which you can kind of see in those pictures that I showed of the Philippines, let's see if I can quickly go back there, is sedimentation, which is a problem that is relatively equivalent to what happens in, so in this picture you can kind of see it. It's equivalent to what, can you put the presentation back on? There you go. So that rock in the left here, it's covered with this very fine gray sediment, and there's a quarry right by those reefs in the Philippines with sediment that runs off when it rains. The shallow reef is not very affected by that, mostly because there's a current that swipes through it, but the deeper reef is more still, so the sediment sits on it. So I think it's more impactful than the shallow reefs in terms of both sediment and hurricane stirred up sand, but eventually they recover. So one of the things we did when we went to the Bahamas was we went down exactly over the track of the hurricane, but then coming back up through the chain, we kind of went out maybe 50 to 100 miles out of the track of the hurricane, and those reefs looked normal. They looked colorful, they were vibrant, they were okay looking. So I think they recover, but we don't know how long it takes. I assume it's slower than the shallow reefs, so that's why I think those impacts are more pronounced in deeper reefs than they are in shallow reefs, but we don't really know. I mean, we don't know the species that are there. So it's just an emerging science. Yeah, there's one here too. There's another one there.
Hello.
Okay.
At the beginning of your, thank you for the presentation, by the way. It's very interesting. At the beginning of the presentation, you show a slide about the Great Coral Reef.
Uh-huh.
And if I'm not mistaken, I got the feeling that there were a lot of more green dots on the right side of the slide.
Right.
What is changing that is actually improving the health of the reef?
So it's not improving the health. Let me go back to that slide and I can explain to you exactly what happened. It's just that in the second event, it was not as widespread. So in the first event in 2016, the temperature was much higher. The pool of hot water that sat on top of the reef was much bigger. So it affected a bigger portion of the reef. So then there was a winter, there was no bleaching, and then the next summer came, summer 2017, and there was another bleaching event. The bleaching event in 2017 was relatively smaller. So it's not, so the reefs that are green on the right and red on the left, it's not that they recovered, it's that they were not affected by the second event of bleaching. So the second event was slightly smaller than the first one. With the Great Barrier Reef, what happens is it's not the entire region that gets warmed. It's usually pools of warm water that are driven in by the currents, and sometimes they sit on top of the reef for too long. But sometimes, I think in the 2017, if I'm not mistaken, the whole event was cut short because there was a typhoon that came. And then it swept through the Great Barrier Reef, which is a different kind of impact, but the typhoons, they help against coral bleaching because they suck in temperature from the ocean. So they feed on, they get grower as they go through warm water, and they make the water cooler. So if they decrease the temperature of the water, then there's less bleaching. Yeah.
Thank you for your talk. What happened to the fish if the coral reef is dead? I'm wondering that.
That's a good question. So yeah, if the species depends directly on live coral, which a lot of species do, they just disappear. There's a lot of butterfly fish, for example, that feed exclusively on live coral and on certain species of coral even. So if the coral species that they feed on disappear, the fish goes together with it. But the most pronounced impact, even when not all of the coral dies, we see a shift in community. So the same way that the community structure shifts from deep to shallow, when you have a bleaching event like that, and you go back the next year and you do a fish survey, the community of fish is completely different. And most of the time, if the coral dies, the diversity of fish decreases. So not only species that dominated before are not dominant anymore, but the number of species in general also decreases because there's less niches. So there's less things that fish can do on the reef because there's less food and there's less diversity, and lower diversity of corals generates lower diversity of invertebrates, that generates lower diversity of fish, and it just cascades down through the whole ecosystem. But in general, it's bad for the fish community.
Okay, I think we have time for one last question. Go ahead.
Okay, I have a question. Okay, in forests, fires are natural events that actually help the forest to stay alive.
Yeah.
Is coral bleaching a natural event that helps the coral reefs to stay alive? Because coral bleaching has been around for probably a very long time.
Yeah,
right?
it has, but not at the magnitude and the frequency that it's happening now. So corals are a very good animal to kind of study those kind of things because they have a hard skeleton, they leave a record behind. So you can go to the Great Barrier Reef and there's these massive, giant brain corals that are 1,000, 2,000 years old. You can reel through them and you can see the record of temperature through time. Looking into the rings, they leave rings behind, just like the trees leave rings behind. So we know that there's bleaching is relatively a natural occurrence that occurred before, but what we've seen in the past 20 to 30 years is that those bleaching events, they're becoming more widespread, so they're much wider than what they used to be, and they're becoming more frequent. So I think the first well-documented global bleaching event was in the mid-90s. And Palau was widely affected by that, and it kind of recovered later. But now there's a global bleaching event almost every year. And so back then, there used to be one every 5 to 10 years. So I think the next one after the one in 97, 98 was 2005, 2006. So it's like 7 years or so. And now it's every year it's happening. This one in the Great Barrier Reef was 2 years in a row. It was the first time that a bleaching event was recorded 2 years in a row. So even though it happened before, it was not in the magnitude that was happening now and not in the frequency that is happening now. So I think, and the follow-up question to that is what's going to happen to coral reefs in the future. There's a lot of people that think that the entire ecosystem is going to collapse and coral reefs are going to cease to exist. I'm not that pessimistic about it. I think there's still a lot of species that are resistant to it. And then even within species, there's individuals that are resistant to it. So I think eventually they will adapt and coral reefs will change. In 50 years, they're definitely not going to be the same. The species that dominate now or that dominated 10, 20 years ago, they're not the ones that are going to be dominating 50 years down the road. But I think the ecosystem as a whole will be there.
All right. On this note, I'd like to thank Luiz again.
Thank you.
Ask you to stay on stage for this.
My pleasure.